Hi, everybody. I just want to tell you a little bit about the exercise we're going to be doing for Unit 3. So we're going to be looking at deep coral communities, and this exercise is adapted from an exercise developed by the National Marine Sanctuary System, which is part of NOAA. Okay, so let's go on. I think this is a really fun exercise. We get to watch some videos, play with some data, think about benthic communities, which of course I really geek out on. So the National Marine Sanctuary System includes a variety of places where we've put aside reserves to protect marine biodiversity. And so these are found all around the United States and its territories, or at least in Hawaii, I guess, American Samoa. So there's also one marine sanctuary in the Great Lakes, a little bit of a misnomer, but cool. All right, so we're going to look at marine sanctuaries along the Pacific coast of the United States for this exercise, including these Greater Farallones, which I still haven't learned how to pronounce. That's on my list of things to do, and the Channel Islands. For this exercise, I also have videos from the other Pacific marine sanctuaries, but we're not going to actually use them for data. But I would encourage you to look at them if you want, just because you'll see some things you don't see in the other videos. So these corals, these deep sea coral communities are found generally in the mesopelagic zone. So these are different than the corals that you're used to thinking about, which mostly occur in the sunlight zone. And we'll talk about the differences here in a moment. Okay, so deep sea coral communities along the West Coast and throughout the world provide habitat for all kinds of organisms, and they occur at depths of about 50 meters to 2,000 meters. The sites that we have videos from for this exercise come from about 150 meters to, I think, 16 or 1700 meters. So still pretty deep. Okay, let's keep going. So our goal with this exercise is to collect data from videos, from these video transects at a variety of locations and depths. And the idea is we'll look at various descriptors of communities, so richness, abundance, and similarity between sites, and see what this tells us about the abiotic and maybe biotic characteristics of these communities. So we're going to use 9 transect videos for this exercise. So I'm going to have you collect data from 9 transect videos. There are 15 transect videos uploaded on our WyoCourses, or rather on a Dropbox that you can access through WyoCourses. We used all 15 last semester, but I'm doing things a little bit differently this time. So we're going to only use 9 of them to focus on. Each transect is about 3 minutes long, so they're relatively short. And your main goal is going to be to characterize the habitat, but also to try and count the different organisms you see, the different taxa that you see, in order to come up with data to do these community analyses. One of the reasons that I've cut down on the number of sites is I want you guys to spend more time looking at these videos carefully and checking your data. So there was a lot of variety in data last time we did this. I think that by double checking stuff and also me providing a little bit more information, you might have a better time getting good data for this exercise. So the habitat types are mostly going to be this rocky bottom, provides lots of structure for different organisms. We'll also look at one video with a soft bottom community, which is pretty cool. This is the kind of system that I've studied. We won't see everything here. Some things are buried in the sediment, so they're not visible, which is one of the problems with this approach, perhaps. There is also one video of a ledge community, which is a sort of cool place where there's this vertical rock face that organisms have colonized. Okay, so deep sea coral communities are composed of both hard or stony corals and soft corals. And they both provide habitat and shelter for different organisms. Deep sea corals are found in all kinds of waters, especially cold waters. Cold waters is deep, like cold as -1. So you might be asking yourself, how does seawater get that cold? Because of salt, it doesn't freeze at 0 degrees Celsius like fresh water does. There are more than 3,000 species of deep sea corals. And some of them are extremely long-lived, thousands of years in some cases. And one thing to think about is these corals are not individual organisms, right, but colonies of individuals. So an individual organism probably doesn't last for 4,000 years, but the colonies might last for a very long time. So to contrast deep sea corals with the corals that you're more familiar with, the warm water, sunlight zone corals, These corals, right, the sunlight zone corals, as many of you probably know, possess algae. They have symbiotic algae that allow them to get energy from photosynthesis from these symbiotic algae. Of course, deep sea corals, where there's less light, don't have this advantage, right? But they instead capture food with their polyps, right? So the polyps, these little individual organisms, have tentacles and they can capture floating zooplankton and other materials that might be drifting in the water column. And it's important to note that these warm water corals do this too, but they also get energy from photosynthetic symbiotes. So again, when we talk about corals, corals are actually generally colonies of individuals. And you can see here with the sea pen, which is a type of coral, all the individual polyps with their tentacles extended trying to collect food on the left. And then on the right, we can see the skeleton, right, of the sea pen, which in this case is not hard, but it's made of organic material, in which the polyps have retracted, right? They're hiding from predators or something. Okay, so we have hard corals, right, which are corals that secrete calcareous skeletons. And this is like the corals you're more familiar with, right? And in these videos, you'll see that some of these cup corals here on the left, I'm trying to remember what they're, Flabellum genus. We probably won't see very many of these, at least not in the videos you're assigned to collect data from. They're super abundant in one of the other videos. And then there's also Lophilia. You will definitely see a lot of these soft corals in the videos you look at. And so the soft corals include the sea pens, like we saw a few slides back, but also these sea fans, which are also called gorgonias. And there are several different species. They're mostly named after their color. But when you're looking to identify them, also pay attention to the morphology, right? In certain lights, these orange and, or golden rather, golden and bubblegum corals can look really similar. But if you look at their morphology, they are different, right? The color looks similar sometimes, but their morphology is different. We might also see sponges. In particular, there are basket sponges and barrel sponges in pretty good abundance in some of the videos that I have looked at. I'm not sure if they're the videos that we're assigned to, but I think you'll probably see some of these. And then, of course, a variety of mobile invertebrates. And one important note here is that both sponges and corals are also technically invertebrates, right? Animals without vertebrae. But we're sort of separating them from these mobile critters like this squat lobster. And I like all these organisms, but this basket star is super cool. I've caught these before. The sun star, these feather stars or crinoids, and then also sea cucumbers, which are pretty abundant in some places. You just have to get your search image down for them. Finally, we have fish. This is where I think a while back I talked about we're looking at taxa. It's more than just species. The rockfish, there are several different species you'll see in the video. For our purposes, don't worry about identifying them to species, just call them all rockfish. And then there are 3 other species of fish that you see a lot less frequently. So these rockfish are fairly common in some videos. The lingcod and these other 2 fish are less common and only found in some of the videos. Okay, so finally, one thing you'll notice in a lot of these videos, or all of them hopefully, are these 2 points of light, laser points. These are basically just scales so you can estimate size of organisms. So they're 10 centimeters apart. So based on that, we might guess that this rockfish is maybe 15 centimeters in length or something like that. Okay. There's the ROV deployment video, which is sort of fun to watch. Gives you an idea what this submersible looks like, what this ROV that's collecting all this images and video looks like. So that's sort of cool. And then, so what you're going to do is, again, each team should analyze the 9 videos listed in the exercise description. This includes 6 transects from the Channel Islands and 3 transects from the Greater Farallones. I'm going to figure out how to pronounce that. You may wish to have different team members specialize on particular groups, right? So maybe you'll be the soft coral, sea fan person, I would suggest that a couple people focus on each group, right? So that you can check each other. And that gets to my last point. You'll probably want to watch the videos a couple times and then double check each other to make sure that you're getting as good data as possible. Again, last time we did this, the data was pretty variable. One of the other things I'm going to do this time that I didn't do last time is give you a list of species you'll expect to see in each video. I think this will help you sort of to use the IDEA guide more effectively and get better data. So that's it. I hope that this is helpful and I hope you guys have fun with this exercise. I think it's really cool looking at all this video. All right.